Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 22nd September 2020. And you may recollect from your memory that we have been talking about generation and conduction of nerve impulse. So we were talking yesterday that when neuron is not conducting any impulse, that is when the neuron is at rest, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. So similarly, we were also talking that the sodium-potassium pump, which transports 3 sodium ions outwards and 2 potassium ions into the cell. And also we were talking about the resting potential, where the cell becomes polarized state. We were talking about the action potential, where the membrane is said to be depolarized, and we were also talking about repolarization, and the action potential jumps from one node to another node and passes along the myelinated axon faster than the series of smaller local currents in a non-myelinated axon. This type of conduction we call it as saltatory conduction. Now we will talk about the next one, that is chemical synapses, that is conduction of nerve impulse through chemical synapse. The junction between two neurons, as all of you know, is synapse, and across which the impulse passes from one neuron to the next neuron is synapse. This is synapse area. This whole thing is synapse area. A typical synapse consists of a bulbous expansion of a nerve terminal called presynaptic membrane. Look at this, presynaptic membrane. And this presynaptic knob lying close to the membrane of a dendrite. So next one is a dendrite. The cytoplasm of the synaptic knob contains numerous synaptic vesicles, as you are seeing. See, so many synaptic vesicles are present. Each vesicle contains a very important liquid called neurotransmitter. Look at this, neurotransmitters will be present. Like we said, acetylcholine, chemical substance. The membrane of the synaptic knob nearest to the synapse forms the presynaptic membrane, and the membrane of the dendrite is called postsynaptic membrane. So you got two membranes, presynaptic membrane and postsynaptic membrane. These membranes are separated by a gap called synaptic cleft. So you got two membranes, presynaptic membrane and postsynaptic membrane. In between the two membranes, the space, very small minute space is there. That space is called synaptic cleft. The postsynaptic membrane contains receptor sites. So on this membrane there will be receptor sites here, and these receptor sites, what do? They receive neurotransmitters. So the two main neurotransmitters in vertebrate nervous system are acetylcholine, we say ACH. So the neurotransmitter is ACH, acetylcholine, and noradrenaline. I will write noradrenaline, Na. Although there are many neurotransmitters that exist. So main thing, at least you should remember two, acetylcholine and noradrenaline. So these are the two neurotransmitters that will be present in these vesicles. When the impulse arrives at a presynaptic knob, when the impulse arrives at a presynaptic knob, calcium ions, we were talking about it yesterday, Ca+, from the synaptic cleft enter the cytoplasm of the presynaptic knob. The calcium ions cause the movement of synaptic vesicles to the surface of the knob. The synaptic vesicles are fused with presynaptic membrane and get ruptured. As you are seeing here, see, it is getting ruptured and ready to rupture. All the vesicles are coming and having their place on the presynaptic membrane. Once the K+ ions concentration goes up, immediately the calcium ion, Ca2+ ions goes up, the vesicles start rupturing. And once they are ruptured, what is that? They discharge their content. What is the content? It is the neurotransmitter. So neurotransmitters will be discharged into the synaptic cleft. So all the neurotransmitters from the vesicles through presynaptic membrane, from the axon of one neuron to the dendrite of another neuron will be entering. So the neurotransmitter of the synaptic cleft binds with protein receptor molecules on the postsynaptic membrane. So on postsynaptic membrane there will be protein receptors, protein receptors. This binding action changes the membrane potential of the postsynaptic membrane, opening channels in the membrane and allowing sodium ions. Now the question is sodium ions. Allowing the sodium ions to enter into the cell. And entry of sodium ion into the cell causes depolarization, as I said yesterday, depolarization and generation of action potential in the postsynaptic membrane. Thus, the impulse is transferred to the next neuron. So this is how the conduction of nerve impulse will take place, and then the nerve impulse not only generates or conducts, it also passes on. So this is how from one neuron to next neuron, through this neurotransmitters in the form of chemical way, impulse will be generated and transferred from one to another. So this is what exactly the nerve impulse, generation and conduction of nerve impulse will take place in two forms. One is through nerves, through neurons, and second is through chemical conduction of nerve impulse. So once you understand this, then we go to the tomorrow session, central nervous system, and we will try to study and understand our own brain. Until then, have a brain.